Physiological death signals could be the life to cancer patients, but we need your help. Here's how. Prostate cancer is the most common cancer to affect men, and is one of the largest causes for cancer-induced deaths. It is a disease of age, with more than 90% of prostate cancer cases being diagnosed to men over the age of 60. Prostate cancer occurs when normal prostate cells accumulate genetic damage, causing them to morphologically change and proliferate into cancer cells. These cancer cells may go on to become malignant and affect distant organs. One factor that affects the formation of prostate cancer is the cancer cell's ability to resist cell death. When normal cells are subjected to internal or external stress, the cells are able to detect the damage and subsequently activate apoptotic pathways, which cause the cell to undergo highly regulated death. When these detection or signaling mechanisms no longer function, you no longer have a way to initiate apoptosis of these cancerous cells. The BCL2 family of proteins is known as an important gatekeeper to the apoptotic response. This group of structurally related proteins comprises of pro-apoptotic and anti-apoptotic members that interact with one another to regulate the initiation of apoptosis. Short sequences of amino acids common to BCL2 and other members of this protein family are known as BCL2 homology domains. These domains, in part, contribute to the function of each member. The BCL family members can be classified into three functional groups: anti-apoptotic proteins, pro-apoptotic effectors, and pro-apoptotic activators. Pro-apoptotic effectors, such as BACs, are closely associated with the mitochondrial membrane and contain BH1, 2, and 3 domains. When stimulated by pro-apoptotic activators, they promote the formation of pores in the mitochondrial membrane, initiating the apoptosis. On the other hand, pro-apoptotic activators, such as BAD, contain only a single BH3 domain. They act by stimulating and activating the pro-apoptotic effectors. Apoptosis-promoting effects from both effectors and activators are inhibited by direct interaction with anti-apoptotic members, such as BCL2 and BCLXL. These proteins sequester BH3-only activators and prevent them from interacting with the pore-forming effectors. These anti-apoptotic proteins can also directly influence effectors to prevent mitochondrial pore formation. Interaction between the anti-apoptotic and pro-apoptotic members is mediated by the heterodimerization of antagonizing BCL2 family proteins. As protein expression of the pro-apoptotic members increase, the response of the cell shifts towards initiating apoptosis and vice versa. This dynamic balance that occurs between anti-apoptotic members and pro-apoptotic members ultimately determines whether the cell initiates apoptosis. Previous research has shown that whilst anti-apoptotic members of the BCL2 family are not normally expressed in healthy prostatic tissue, it has been found that they are extensively expressed in prostate cancer cells, particularly in the non-androgen dependent stage of prostate cancer. Whilst pro-apoptotic members have also been found to be downregulated in cancer cells, studies have shown that an increased expression of anti-apoptotic family members is enough to disturb the dynamic balance that determines the initiation of apoptosis. Hence, in prostate cancer cells, an increased expression of anti-apoptotic proteins, as well as a decreased expression of pro-apoptotic proteins, will lead to an inhibition of apoptosis.
By targeting both anti-apoptotic as well as pro-apoptotic proteins of the BCL2 family, we can induce the prostate cancer cells to undergo apoptosis. Hence, we propose a novel therapy that consists of 4-arrow-4H-chromin derivative as well as an upregulator of oxidol nitrodomain containing protein 1, NOR1. 4-arrow-4H-chromin derivatives, more specifically 3-NC, are novel anti-cancer agents which have been shown to have pro-apoptotic effects in several cancer cell lines. 3-NC has been shown to downregulate BCL2 expression whilst upregulating BAX expression. Presently, no research has been done on 3-NC's ability to work in prostate cancer cell lines. However, previous research has shown that other 4H-chromins have been successful in targeting prostate cancer cell lines. NOR1, on the other hand, is a novel tumor suppressor gene whose apoptotic activity has been demonstrated in various cancer cell lines, including prostate cancer. It works by upregulating pro-apoptotic BAX and BAC and downregulating anti-apoptotic BCL2 and BCLXL. Studies have shown that the expression of NOR1 is decreased in prostate cancer cells compared to normal prostate tissue. By upregulating NOR1, we can effectively increase the ratio of pro-apoptotic members to anti-apoptotic members, ultimately causing cell apoptosis. We believe our novel therapy, by targeting BCL2 family proteins, can inhibit the progression of prostate cancer and effectively eliminate cancerous cells.